Now we're going to get into the classification of microorganisms. But before we do, the general knowledge is that all living things in this world are divided into 6 different kingdoms as illustrated in this figure here. Except for these 4, animal and plant are not considered as microorganisms because they have tissue differentiation, which basically means that tissue differentiation leads to formation of organ, and then those organs will form higher organism, which microorganism doesn't have that. Given that plant and animal are typically large in size, so those can be seen by naked eye. However, for microorganism, microscopic life is technically really tiny. We need the aid of microscope to see them. So those microorganisms actually are spread into the rest of the kingdom: Eubacteria, Protista, fungi, and Archaebacteria. In terms of the complexity of the organism within microorganism group is that Archaea make the simplest microorganism, and fungi is the most complex microorganism within a microbial group. Among these 4 kingdoms, each kingdom poses specific characteristics, and some of the characteristics actually being shared between kingdoms. For example, Eubacteria and Archaebacteria are prokaryotic cell, whereas fungi and Protista are eukaryotic cell. We will explain this later in the following slide. But if you see this figure, you don't see virus belong into any of this group. So my question would be, how about virus? Is it a living thing? If it's not a living thing, why? I will let you to find out about that, okay? Here's the anatomy of eukaryotic versus prokaryotic cell. I believe this is not a new knowledge to you, but let's refresh some of the key important points. Let's highlight the differences between these 2 cells. For eukaryotic cell, the genetic material is multiple linear and closed within nucleus. All the organelles for eukaryotic cell have its own membrane, where prokaryotic cell doesn't have that. Eukaryotic cell doesn't have cell wall, but some of them do. However, for prokaryotic cell, they do possess cell wall, and some of the prokaryotic cell actually have another structure, the most outer layer structure called capsule, which serves to protect the prokaryotic cell from severe condition. Prokaryotic cell usually pretty simple, so the structure is usually smaller. The size is usually smaller than eukaryotic cell. Between these 2, they also pose some similarities. I would like you to find out all the similarities between these 2 cells. So let's get started with the first group of microorganisms, which is also the prominent one, is bacteria. So generally, bacteria require oxygen to grow, but some of them prefer less oxygen content to grow, which is called anaerobic microorganism. Most of them prefer pretty neutral pH condition. In terms of temperature, the optimal temperature growth for most bacteria is around 37 degrees Celsius. However, some microorganisms are psychotrophic, meaning that it can multiply in a refrigerated temperature. So that's why like milk, for example, we have to put it in the refrigerator after we open it, but it only lasts for several days, because after we open it, we already exposed to air, so it only have certain number of days to make sure that it still can be safe to consume. Some bacteria can be motile with the aid of flagella, so they can move using this flagella structure. Bacteria also differ in shape. Typically, rod-shaped bacteria is called bacilli. Round, grape-like structure shape is called cocci, and also like a corkscrew shape is called spiral. Most bacteria can be grouped into 3 major ones, The first one is harmful bacteria, spoilage microorganism, and also pathogenic microorganism. So these two types of bacteria group, spoilage and pathogenic, is the most concerned one that can cause us sick and sometimes even death. Another type is harmless, or known as commensal bacteria. It's just abundance in the environment, is naturally present in the environment. The one that we use it for our benefit is actually lactic acid bacteria. So if you remember, yogurt is one of the fermented food where we use lactic acid bacteria to ferment the milk to form the yogurt. So we know that probiotic in general help to improve our digestion system. So there's a bacteria that we can use for our benefit. Here's just showing you different types of flagellar arrangement on bacterial cell. One of the examples that I want to highlight here is Vibrio cholera, where it poses only one flagella at one end. Most foodborne pathogens have protrusive flagellar arrangement like this, where there's multiple flagella around the edge of the cell. E. coli, Salmonella typhimurium, Yersinia, and Bacillus is some of the examples. For Pseudomonas, which is one of the spoilage microorganism, it has a multiple flagella on one end. So this is called low protrusive flagellar arrangement. Bacteria can also be classified based on cell wall structure. Remember, bacteria is prokaryotic cell, and prokaryotic cell has cell wall. So one of the important technique in microbiology is actually gram staining technique, where it differentiates between gram-positive and gram-negative bacteria. The difference between these structure is that gram-negative has a very thin layer of peptidoglycan, whereas for gram-positive bacteria has a very thick peptidoglycan. After gram staining, the result under microscopic observation is that gram-positive bacteria will appear as purple in color because the crystal violet stain remains. And one of the examples of gram-positive bacteria is Staphylococcus aureus. For gram-negative, it doesn't retain the purple color, and after this stain, it will pick up the saffron in color, which is red in color. So it remains red after gram staining process. Some bacteria, such as Bacillus cereus, has endospore, and it can be observed after performing endospore staining. So if you can see in this figure here, the green structure here is actually the spore. Instead of using crystal violet for endospore staining, it uses malachite green stain at the first step of the staining process. So basically, this figure here just summarizes the gram stain procedure. For your concern, we don't have to get into this technical, but this is for your information. And also, this is one of the important techniques for microbiologists. Another classification can be based on gases requirement for microbial growth. So here are 5 different types of gases requirement in terms of the need of the oxygen. So the first one is obligate aerobes, mean it needs oxygen no matter what. That's why if you grow the bacteria in the tubes here, it always stay almost at the surface of the media. The second one is obligate anaerobe, which means that it will grow without the presence of oxygen because oxygen can kill them. So that's why in this tube here, it will stay at the bottom of the tube to avoid oxygen on the surface. The third one is facultative anaerobes. It does not need oxygen to grow, but a little bit of oxygen will not harm them. For the next group is actually macroaerophile, which means it needs a very low concentration of oxygen to grow, just a little bit. So that's why it usually grows little under than the surface of the media. The last one is aerotolerant. It does not need oxygen to grow, but it affected by it, means it will grow in all over of the media because oxygen is not a concern for them. This is another figure that also map different characteristics of bacteria based on shape, based on cell wall, based on gaseous requirements. So actually, rod-shaped bacteria is actually the structure for most foodborne pathogens like Vibrio, Campylobacter, Shigella, Salmonella, which is prominent one, Pseudomonas, Listeria, Bacillus, Clostridium. So based on this figure here, you can spot the characteristics of each bacteria. Like for Bacillus, it is spore formers, it has spore. That's why endospore staining can be used to visualize the spore. It is gram-positive and it rod-shaped bacteria. So that's just one of the examples. By now, you may have heard me saying foodborne pathogen all the time. So what does actually mean? So foodborne pathogen. So pathogen basically refers to harmful bacteria. And foodborne means that harmful bacteria, or better term as pathogen, has to be present on the food before it gets transferred to human as a vehicle to get sick. So that's why foodborne pathogen term is always used when we describe about these example of microorganisms such as Salmonella, Staphylococcus aureus, Vibrio, as well as Clostridium botulinum.